Hey, welcome to In the Lab with Leo. I am Dr. Leo Chen, your lab buddy. Today we're going to talk about the Annexin-5 PI assay. The Annexin-5 assay is one of the most commonly conducted apoptosis assays. The oncology and immuno-oncology researchers routinely perform Annexin-5 assays during the development of novel therapeutic treatments. Because Annexin-5 is an early stage apoptosis indicator, when staining cancer cells with Annexin-5, researchers will be able to identify and characterize the state of the cells after certain treatments. When apoptosis is triggered by the extrinsic pathway, one of the first physiological changes that occur within the cell is the externalization of phosphatidylserine, or PS, an anionic phospholipid, from the inner leaflet of the cell to the outer leaflet. When this occurs, fluorescently labeled Annexin-5 can bind to PS residue on the outer leaflet and emits fluorescent signals showing that apoptosis is in progress. As the cell progresses through various stages of apoptosis, it's inevitably headed towards cell death. And one of the hallmarks of cell death is the irreversible permeabilization of the cellular membrane. And at this stage, propylium iodide, or PI, can enter into the cell and bind to the DNA. So what's the importance of the Annexin-5 PI assay? Now, within a single experiment, immuno-oncologists can determine the efficacy of a potential cancer drug treatment by simply looking at the population of cells that are stained with Annexin-5 and or propylium iodide. While this seems pretty simple, there's always a few things to keep in mind. So here's my tip of the day. Please always remember that apoptotic cells are fragile. If you pipette them too hard, they may break apart. If you centrifuge them too hard, they may disintegrate. And of course, keep an eye on your palate. Post-centrifugation can be very tricky. It is easy to accidentally pipette off some cells, and those cells may very well be the apoptotic cells that you're looking for. Next time, we're going to be talking about caspase-3, a late-stage apoptosis assay. Hey, if you like this video, please subscribe to our channel for more content. Check out our website in the link below. Follow us on Twitter, Facebook, and LinkedIn. I'm Dr. Leo Chen. See you in the lab.